Welcome to Let's Get to the Marks, going through A Level Biology. This is the 2024 Paper 1, 10 questions, 2 hours. Make sure at the start of each question you pause the video, have a go with a pen and a paper, and then unpause and watch the walkthrough. All right, I'm going to go through the explanation. So figure 1 shows the structure of a fatty acid molecule and the structure of a glycerol molecule. Which is which? Ooh, you should be able to work that out. On figure 1, draw a circle around the part of the fatty acid molecule and the part of the glycerol molecule that is removed to form a triglyceride. Name the bond formed between the fatty acid and the glycerol in a triglyceride molecule. Name the reaction involved in forming a bond between the fatty acid and the glycerol. Let's shrink this down so we can get these answers down. So, yeah, like I say, pause, have a go, and then unpause, and we'll go through it. Okay, so first off, we have which is which. Well, actually, this is propan 1,2,3 triol for you chemists, or glycerol, as it's called in biology. And this is the fatty acid. Acid because it's got this carboxylic acid group, the COOH. Now, you can remove the OH from the carboxylic acid and an H from one of the alcohol or OH groups on the glycerol. But we can do it the other way around as well. We can remove the whole OH from the alcohol and just one H from the carboxylic acid, as long as you're removing two H's and an O. And then what's going to be left over? What is this type of bond called? Well, let's draw it out. So imagine you snap off that H, you snap off that OH. What are you left with? Well, you're left with the C double bond O, and then you have an O, and then a C, because the H and the O have gone. And this is the ester link or ester bond. And if you're a chemist, you know between carboxylic acids and alcohols, you get an ester bond. Okay, and the type of reaction, well, you've removed two H's and an O, H2O, that's water you've removed, so you see water form as a product. That is condensation. Sorry, I just bit my tongue there. Okay, let's move on. Table 1 shows information about 3 fatty acids. Okay, so we've got a little table here, and it's got the diagram of each fatty acid structure, known as the displayed formula. If you're a chemist, name of the fatty acid, we've got stearic, oleic, and linoleic acid. We've got fatty acids melting point, so it goes 70, 14, and -5. So what's going on here? Name the fatty acid that is a saturated fatty acid. So what we want to do is look for any double bonds, because if you've got a double bond, you're unsaturated. Now there's one here, so that's mono, and this one has two, so it's polyunsaturated. And then we can see the saturated one, the top one, stearic acid, doesn't have any carbon-carbon double bonds. So we're just going to put stearic acid. If you had to explain, you'd say it's saturated because it has no carbon-carbon double bonds. The melting point is the temperature at which the solid changes to liquid. Use the table to describe the relationship between the fatty acid structure and the melting point. So look, as we go down from saturated to monounsaturated to polyunsaturated, the melting point decreases from 70 to -5. So we could say the more unsaturated or the more double bonds you have, the lower your melting point. That's one way of wording it. So I don't know why I've written fatty acids, but yeah, as the fatty acids become more unsaturated, the melting point decreases. So that's one way to get the mark. Of course, you might see on the mark scheme it says or vice versa. What it means by that is you could argue that the other way, but it means the same thing. We could say as we go up, we could say as we get less unsaturated, or we could say as we get more saturated. So as the fatty acid becomes more saturated, as it has less double bonds, the melting point increases. This means the exact same thing. I've just written it as an alternative. If you're sitting there wondering, is my answer right? You can word it any way you like, as long as it's true. Now, I'm going to go through the explanation. This question doesn't give you an explanation, but there's actually an explanation as to why this happens. So my little rectangle here is a glycerol, and then these are the 3 fatty acid tails you would get on a triglyceride. Now, that is the crustiest little drawing. I did get a level 4 in GCSE art, so you've got to have patience. Now, between molecules, we get intermolecular forces. If you're a chemist, you know this. Unsaturated fatty acids have double bonds. Now, because they can't rotate, there are bends, which we call kinks, in biology, okay? So these kinks in the fatty acid tails mean that the tails spread out. They can't get close to each other. And it means if you have 2 molecules, or if you have 100 molecules or whatever, they cannot approach each other closely and arrange themselves in a nice uniform structure. They have weaker intermolecular forces than saturated fatty acids. And that means they have lower melting points. And so the more unsaturated fatty acids you have, the more fluid, the more liquid it's likely to be. So if you look at olive oil, that's going to have loads of unsaturated fatty acids, hence the kinks. If you look at a solid bit of butter or ghee or lard, it's going to have loads of saturated fatty acids with a high melting point. 1.4, this is a really hard question actually. The ratio of saturated fatty acid to unsaturated fatty acids in cell surface membranes determines the extent of the membrane's fluidity. Scientists provide a cell structure of mouse phagocytes with liquid broth rich in unsaturated fatty acids. The scientists observed an increase in the proportion of phospholipids in phagocytes that have unsaturated fatty acids. So if you have phospholipids, you tend to have unsaturated fatty acids. Suggest and explain why there is more phagocytosis. So essentially, this is a really hard question. I think actually understanding what the hell this question was talking about. It's basically saying link the phospholipids and the unsaturated fatty acids to phagocytosis. And if you remember phagocytosis, I can't speak. Rewind. Remember, phagocytosis is when a white blood cell engulfs a pathogen. And what does it have to do? It has to have a flexible membrane. So you've got to kind of infer all of that from this stem of this question. So the more phospholipids there are in the membrane, it appears that their data shows the more unsaturated fatty acids they had. And that means if you have more unsaturated fatty acids, of course, you've got a lower melting point, as we saw, which means your membrane is going to be more fluid, more liquid, more flexible, more able to engulf a pathogen and do phagocytosis or engulf a cell, more able to form a phagocytic vesicle or phagosome, as you know, depending on your exam board or the book you're reading. So here's a phagocyte, and here's it flexing its membrane around a pathogen and then eventually forming a phagosome or phagocytic vesicle. So you kind of needed to piece that all together from the stem. I thought this question was particularly hard. Let's move on to the next question. Let's go. Welcome to Let's Get to the Marks, going through Transport in Cells, A-Level Biology. Why don't you pause the video, grab a pen and paper, have a go at this question, try and get 3 bullet points about facilitated diffusion and active transport down, and then unpause the video and watch me go through the explanation. All right, I'm going to go through the explanation now. So let's think about facilitated diffusion. Well, what have we got? I've got my little phospholipids in a membrane. I've got this thing here in the middle of the phospholipids with like a hole through the middle of it. What's that? That's a channel protein, if you didn't know. And facilitated diffusion is when substances move from a high concentration to a low concentration through a channel protein. Now, why is it that these particles have to go through a protein? Why can't they cross the phospholipids? Why can't they go by simple diffusion across the membrane? And this is because they are charged particles, or they are non-soluble in lipids. They are polar molecules. They are charged. They are hydrophilic. So they will be repelled by the hydrophobic tails in that bilayer. A real thick bit of that cell membrane is just hydrophobic tails, and they're not going to let charged particles through. So the point we want to get across here for facilitated diffusion is that it's charged particles that are involved, or ions, or polar molecules, and they move from a high concentration to a low concentration across or through a channel protein across a membrane. Now, specifically, the marks on the mark scheme here, they give you 1 mark for saying charged particles. Don't worry if you miss that out. That's probably the easiest mark to miss. You at least get a mark for saying from high to low conc through a channel protein, and so that will get you a mark. You can still pick up 2 on the old active transport bit of this question. So now we're going to look at active transport and think about what is active transport. Well, active transport is the movement of substances across a membrane, a cell membrane, and it's against the concentration gradient. So this time we're going from low to high, and it's via a protein carrier. Now, which of these points are actually going to get you up to 2 marks? Well, you kind of needed to say all of these just to get 1 mark. So you needed to say protein carrier, and you had to say using ATP, and you had to say against the concentration gradient. And so actually, for both active transport and facilitated diffusion, we needed to say that they involve charged particles that can't simply diffuse across the membrane. Now, what I'm showing you here is a protein carrier molecule. It's different than a channel protein. So They will reject channel protein for active transport. ATP being broken down into ADP plus inorganic phosphate, the carrier protein, almost like an enzyme with an active site, picks up the molecule, rotates, which is the bit that needs energy, and then releases that molecule on the other side. And obviously there's some 3D changes in its shape or changes in the 3D structure when it picks up and releases. So, and of course we need to say it's from low to high. So there's my little level 4 art diagram there. All right. Let's go on to 2.2. So really the key here was you were limited to only 2 out of 3 marks unless you mentioned charged particles somewhere. Right, 2.2. What are microvilli? Okay, so you've all heard of microvilli. How would you describe microvilli? Well, that is the crustiest little small intestine I've ever seen. If you zoomed in on the small intestine, you'd see these extensions, these folds in the epithelial layer, in the epithelial cells of the small intestine, which are the cells that cover the linings inside the surfaces inside our body, like our lungs, our intestines, or any exchange surfaces. So we've got these epithelial cells, and they're folded into extensions, which we call villi, extensions of the cell membrane. And then we have further extensions, even more extensions on those extensions, even more folding. And these are the microvilli. So they're even smaller than the villi. Even each villi is further subdivided into microvilli. This gives a large surface area. So what we need to say is extensions on the villi, on the epithelium lining of the small intestine that provide a large surface area. I also need to mention that this occurs on, these are folds on the cell surface membrane. Now, I didn't mention cell surface membrane here, so I wouldn't get the mark. Surface is in brackets on the mark scheme because you don't need to say surface. You can just say cell membrane. It assumes, when there's something in the brackets, it assumes you know that. Okay, so 2.3, vitamin A is a fat soluble substance. My cells are involved in the process of vitamin A absorption. Describe the process of vitamin A absorption into the cells lining the ileum. So we're talking about those microvilli, those epithelial cells lining the ileum, which is a section of your small intestines. Now, why does vitamin A need to go in a my cell? Like other things like glucose, of course, doesn't need to go inside of my cell. Well, vitamin A is actually lipid soluble. It dissolves in lipids. It's a non-polar molecule, and that means it's not very soluble in the solutions and substances moving through our body. So in order to make it more soluble, phospholipids, mainly phospholipids, form a sort of single layer membrane around the vitamin A with the, they'll do this with any fat soluble substance, but vitamin A is just one of those. So it's an example. And these, they're not trees, by the way. These are supposed to be phospholipids with the 2 tails, but they're incredibly crusty drawings. I did tell you before I got level 4 in GCSE art. So these face with their hydrophobic tails attached to the vitamin A because the vitamin A is fat soluble, and the hydrophilic heads facing outwards. And we call this a micelle. So this is a micelle. So I'm going to write my cells arrange themselves or my cells. You don't really need to write all this. You can just say my cells are formed from, but I'm just overdoing it. Phospholipids, um, hydrophobic tails attached to vitamin A because it's fat soluble, and the hydrophilic heads face outwards. And this means it can interact with water and solutions that are found passing through the human body, like the digestive system. Now, also, the micelles combine with bile salts. This makes it even more soluble. Bile salts are charged salts. You think of salts are ionic, so they have like plus and kind of negative charges. So these are bile salts, and this further improves the solubility. So micelles combine with bile salts. The micelles break down. They actually constantly reform, break down, reform, break down. It's a bit random. They're like the phospholipids come apart and the vitamin A is released, then it's bound back up inside the phospholipids, then released, then bound back up, and then released. And eventually it moves towards the cell membrane. So micelles break down close to the cell surface membrane of the ileum. Getting a bit specific there, but we're referring back to the question. So they're going to break apart and they're going to release. So they're going to kind of come apart like this. Let's just draw it like that. I won't overdo it. And then they release the vitamin A. And the vitamin A is now at the cell surface membrane. Yep, there's my really shortened version of the cell surface membrane. Obviously, it's got carrier proteins, it's got glycoproteins, it's got all sorts going on. Anyway, the vitamin A can now simply diffuse across because it is fat soluble. So my cells break down close to the cell surface membrane of the ileum, releasing the vitamin A, which which can now, sorry, every time I do a spelling mistake, I make a stupid noise. It's like become a glitch in the system. Releasing the vitamin A, which can now diffuse, oh my gosh, diffuse across the membrane. I've already said cell surface membrane. I'm not going to mention that again because it's just long. So what do we really get the marks for here? What we're focusing on is the key marking points. So we want to say that my cells and bile salts join together. We want to say that my cells break down close near the cell surface membrane and that the vitamin A diffuses across the membrane because it's a lipid. The membrane is permeable to lipid soluble substances. And hopefully you'll have got your 3 marks. All right, guys, don't forget sharing's caring. Hit the like, punch up the bell, subscribe. Got that all in the wrong order, but it don't matter. Just make sure you like the video and I'll catch you on the next one.
You're still here? It's over.
Okay, time to go through question 3 for the old A-Level Biology Paper 1 2024. So let's get into it. This is for the AQA syllabus. Describe how we breathe in. 3 marks. No explain, just describe the process. So what I'm going to go through is the process. And what you're essentially looking to describe here, I'd pause the video and have a go with a pen and paper if I was you. But this is like the thorax, the upper chest area. And what's going to happen is the pressure is equal from the outside to the inside. What we need to do is make our thorax bigger so that the pressure goes down below 1 atmosphere, which is the pressure outside, and that will cause the air to rush in down what's called a pressure gradient. So what you need to focus on with this question is what physical changes must happen in the body so that we can increase the volume of the thorax and reduce its pressure so we make a pressure gradient. So first of all, the diaphragm must move down or flatten. The diaphragm is the muscle underneath the thoracic cavity. By flattening or moving down, it makes the volume bigger. The next thing to make the volume bigger, you can put your hands on the side of your chest and breathe in. You'll feel your ribs go up, and that's the external intercostal muscles that contract. These move the ribs up, but they also move them forwards as well, out. You can try it. Take a big breath in. There you go. Now, what does this do? What does the ribs moving up and the diaphragm moving down do? This actually causes the thoracic cavity's volume to increase, as you can see by my incredible drawing. Excuse me. So this increases the volume of the thorax or the thoracic cavity, and it decreases the pressure because the bigger the space, the further less frequent collisions between the particles and the walls of the container, which is your thorax. So the pressure goes down and it falls below atmospheric pressure, so air moves in down a pressure gradient. This is an example of mass transport in animals, a way for us to move large volumes of material using some physics. Okay, not using osmosis diffusion or active transport. This is using a big physical process. Okay, and that will get you your 3 marks. Right, let's look at 3.2. A scientist prepared alveolar tissue to view using an optical microscope. That means they cut a slither of someone's lung tissue. The scientist cut very thin slices of the alveolar tissue. Explain why the scientists use very thin slices of alveolar tissue with the optical microscope. So again, pause the video, see if you can write this down. This is more of a GCSE style question. It's quite an easy one. So why do we use thin slices with an optical microscope? To create a thin layer of cells which will make light easy to pass through. So it's really triggering in your mind that optical microscopes don't use electrons, they use light. We want that light to be able to cross through the cells, so you're using a thin layer of cells. 3.3, figure 2 is an image of a lung tissue observed under an optical microscope. So we've got the alveolar epithelium here, we've got these tubes, and they're asking us what is tube A? See that tube just before the alveolar? What is tube A? So I'll go through the explanation, but you can have a go before I go through it. Okay, so if you draw out your basic GCSE diagram, check out this level 4 art. If you draw one of these basic of the lungs, so this is just one lung, you can see I've got the T for trachea. And then the next tube is the bronchi, which has a bit of rings of cartilage, just like the trachea. And then we have the bronchioles, which don't really have any cartilage or much at all. And then we have the alveolar. So what is A pointing to? Well, A is really pointing to the bronchioles, okay, the smaller, tiny tubes. And how do we know that from the diagram? Because they're the thing just before they're labeled alveolar. So if you know the order, trachea, bronchi, bronchioles, alveolar, you know what A is. It's the bronchioles. Okay, let's zing, 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 zing. Right. Oh, a little maths one. The scientists used a ruler to measure the diameter of some alveoli. Table 2 shows the scientists' results. So we've got the alveolus diameter in millimeters. Alveolus diameter 4251235.2. The magnification of the image in figure 2 is times 40. Use this information and table 2 to calculate the mean diameter in micrometers or micrometers of the alveolus. Show your working. So we've got millimeters, we've got micrometers. There's a clash. What equation do you need? You need the I am. Why don't you pause the video, have a go? All right, the tricky bit with this, as I explain it, is going to be working out that this was the I am equation. This would have confused people because they've given you the mean diameter, but they haven't said if this is the actual or this is the image size. But let's face it, you're not going to have an alveoli in your lung. You've got a million alveoli in each lung at least. You're not going to have a 4 millimeter alveoli. These are microscopic, or they're very, very small, tiny air sacs. So they're a lot smaller than 4 millimeters. So that information there with the alveolus diameter in millimeters, that information is the image size. So this is image. So what you're doing here is calculating the average image size or mean image size. So we're going to do that by going 4 plus 2 plus 5 plus 1 plus 2 plus 3 plus 5 plus 2. And then we're going to divide that by 8. Hopefully I wrote those all out. And that comes to 3 millimeters. Now, we need to, at some point, convert our units so they're the same. So I can do it now, or I can do it once I've found out the actual size. So we're trying to discover the actual size. I'm going to times by 1,000 because I want to convert millimeters to micrometers. And so I've got 3,000 micrometers, and I need to divide by the magnification to get the actual size. So 3,000 over 40. I could have done 3 over 40 and then time 1,000 after. I'd have got the actual size in millimeters, but I've done it this way. So, and it's 75 micrometers, and that's going to get you 2 marks. So the difficulty here was really working out that you were doing I am. The clue was that they gave you the magnification, right? And the clue was that they gave you some measurements. So it meant that you had image and something else. It meant you had magnification and something else, and it had to be the image. Right, 3.5. Give the uncertainty associated with taking a measurement using a ruler with 1 millimeter graduations. Calculate the percentage error for making a measurement of 4 millimeters with the ruler. Right, why don't you pause the video, have a go. Okay, I'm going to go through it because there's a little nuance to this, which they have in biology. They don't do this so much in chemistry, but uncertainty, the smallest division divided by 2, that's dead easy, right? Aha, we have a little issue here. We're using a ruler. So when you take a measurement with a ruler, you're going to look at 0 millimeters and then you're going to go up. Oh, it's 25 millimeters long. You're going to, or 5 millimeters long. You're going to look up to another number. So you're actually got uncertainty in 2 places on the ruler. It's the same if you use a thermometer. Whereas if you're weighing something on a weighing balance or a weighing scale, whatever you call it, a mass balance, whatever, you're just going to get one number. So you're just looking at one number. There's only one level of uncertainty. Here, we've got to multiply our uncertainty by 2 once we work it out to get the total uncertainty. So whenever you take a measurement with like a thermometer or a ruler, you are looking at 2 different points, and you're looking, so there is uncertainty at each point. Okay, and so there we go. Yeah, I've over explained that. You get the idea. So we want to do the smallest division. What was the smallest division? It told us the ruler had 1 millimeter graduation. So 1 millimeter was the smallest. We want to divide that by 2, and we're going to get 0.5. So 0.5 is the uncertainty, but the total uncertainty is times 2 is 1. Now we're going to work out percentage error. Okay, I lost my train of thought. My daughter was banging on about octonauts and asking me to put it, whatever the episode with the swordfish on. I'm not really sure what she was on about, but I just had to run off and sort that out. So we're looking at percentage error, folks, and you've got to know the percentage error equation off by heart. The percentage error is your uncertainty, which we calculated in the first calculation, divided by the measurement times 100. So in this case, they've told us you're using the ruler to measure 4 millimeters. We know the uncertainty was 1, the total uncertainty, because in a ruler you double the uncertainty. So it's 1 divided by 4 times 100. The answer is 25% for the percentage error. And there we go. That will get you the marks. Okay, welcome to Let's Get to the Marks, going over a question on biochemical tests. Let's go. Okay, 4.1. Molasses is a solution obtained from sugar beet plants. The sugars present in molasses are sucrose, glucose, and fructose. Give the number of different types of monosaccharides present in molasses. It's worth 1 mark. Grab a pen, pause the video, have a go. Okay, I'm going to go through the answers. So mono means 1, saccharide means sugar. So we're looking at the simple sugars. Now, there are 3 sugars in this, and you need to know your 3 monosaccharides and your 3 disaccharides off by heart. So you should have got these. Your monos are, your monos are, you've got glucose, you've got fructose, and can you shout out the third one? You got galactose. Okay, your disaccharides. So if you don't know these by now, you need to write these on the back of a revision card. They're only 1 or 2 markers out of your whole exam, but it's worth learning them off by heart. You've got lactose, you've got sucrose, as you can see up here in the question. And then what is the other disaccharide? Maltose. And you actually have to know what each of these disaccharides is made of. Anywho, our 2 monosaccharides in this question are glucose and fructose. So this was really just testing you straightforward, AO1, testing your knowledge. Okay, beautiful. Right, let's go down and do 4.2. 4.2, a student used the biochemical test for reducing sugars on a clear sample of molasses. Describe the biochemical test for reducing sugar. Explain the result expected from the test on the sample of molasses. So, do you know the name of the biochemical test for reducing sugars? Hopefully you do. The name is the Benedict's test. And luckily for us, the Benedict's test, it begins with a B. It's always like that. And it's going to turn from blue to red. Or really, it actually goes through all the colors of the rainbow. If you read the rainbow out from red, it's going to go through all those colors in the rainbow before it settles on blue. So all the colors between red and blue. So Benedict's test, it will, what you do with the description of it, you're going to warm gently in a water bath. So that's one of the extra things you normally say about the Benedict's test. It will form now you could say orange, if we go Roy Gabiv through the rainbow, yellow, green, or blue. Sorry. Oh my gosh, what is wrong with me? It will form a green, yellow, orange, or red. Precipitate. Went the wrong way round. So if you think about the rainbow, red, orange, yellow, green, blue, we're going in that way. So it's going to be green, yellow. It's going to go from blue to green, then to yellow, then to orange, then to red. Anyhow, what the hell am I waffling about? You could literally just say red precipitate, but I think they give you the mark for any of these colors because it can legitimately go those colors. And then the name is the Benedict's test and it will form a red precipitate. And Benedict's test begins with B, and it is blue to start with. Now, the explanation of the result. Why did it change color? What is it out of these 3 that made it change color? So out of the glucose, the fructose, and the sucrose. Well, it's because glucose and fructose are reducing sugars. So glucose and fructose are reducing sugars. Now, just for your own knowledge, it's not part of this. That will get you the mark. Glucose and fructose are reducing sugars. I could say so they react with Benedict's reagent or solution. Now, actually, just for your own knowledge, because you need to know this as well, sucrose is a non-reducing sugar. This requires hydrochloric acid and then sodium, like hydrogen carbonate, any sort of alkali or base to neutralize the acid first, then the Benedict's test. So it's slightly different. The test for non-reducing sugars requires you to add acid first, then neutralize the acid, and then you can go ahead and do the Benedict's test once you've hydrolyzed the bonds with the acid and neutralize that acid. Okay, Coolio. And let's look at 4.3. 4.3, free sugar is the sugar in food and drinks released when food is crushed or when sugar is added to food at home by manufacturers. Scientists recommend that no more than 5% of the energy consumed per day should come from free sugar, so added sugar to your meals. The mean daily energy requirement for a 10-year-old is 8,000 100 kilojoules. The free sugar in 1 tablespoon is 250 kilojoules. Calculate the number of tablespoons of molasses required for a 10-year-old to reach the recommendation for energy consumed in free sugar per day. So how many tablespoons of sugar will get them to the maximum added sugar you should have, including all the chocolates, crisps, and whatever else you eat? Right. Well, first of all, we need to find out how many tablespoons are in that. There's a number of ways to do this. And then they're only allowed 5% of that. So that's the total energy per day, and then 5% of that. So we could do it in two different ways. So we could go, okay, I want to do 5%. Now, how do you do 5%? I'm writing 0.05. Well, if you want to multiply something and find out how much energy a child should have a day, it's 5% of 8,100. So I could do 5 over 100 times 8,100. And this is going to give me the first part, but it isn't going to get me the mark yet. So we're going to go 0.05 times 8,100, and that is going to be 405 kilojoules. That's how much energy can come from sugar for a 10-year-old. Now, how many tablespoons would that be? Remember, each spoon contains 250. So we're not even going to get 2 tablespoons here. We're expecting an answer less than 2. So we're going to divide that by 250. And what are we going to get? We're going to get 1.62 or 1.63. It's up to you how you do this. I'm going to put 1.62 on my calculator. So there we go. Wunderbar. And let's give ourselves a mark and move on. Right, we're on to 4.4. A scientist used the apparatus in figure 3 to investigate osmosis. We've got air at the top of a tube. We've got molasses, which is that sugar concoction of sucrose, fructose, and glucose. And it's in a beaker of water with an animal's bladder at the end of this apparatus, I guess, preventing the sugars from leaking out. Use your understanding of osmosis to explain why the air pressure in the tube increased. It's 3 marks. Why don't you pause the video, grab a pen, grab a paper, have a go, or just do it in your head? And now I'm going to go through the answer. Okay, so if you think about, we've got some air in this tube. They're asking us specifically, why does the air pressure in here increase? So why does the air pressure in here increase in that little bit there? So what's going to happen? The molasses or the solution is going to go up, right? And that is going to reduce the volume, meaning the air particles in there have less space and they're going to collide more, increasing the pressure. But they want our biological, talking about osmosis reason as to why that volume went down and increased the air pressure. So what must have happened to force this up? We've got water outside, sugar inside. Well, water moves from a high water potential to a low water potential. So water is going to want to come into this sugar solution. Now, it can't get through the glass, but it can get through the animal bladder. Now, you might say, well, how am I supposed to know that? Well, an animal bladder is made of animal cells and it's going to cross through a cell membrane. Think of your water potential business, your osmosis. So we've got a high water potential into a low water potential by osmosis through the animal bladder, which is a partially permeable membrane. This raises the molasses solution up, decreasing the volume, increasing the air pressure. Let's put that in words for you, and then we can go. Right, so the water moves into the molasses solution through from, let's say, first of all, from a high water potential. Well, you can say a less negative, but we're going to go from a high water potential to a low water potential across the animal bladder, which is a partially permeable membrane. So, this raises, oh my days, raises the level of the molasses solution, meaning a smaller volume of air and therefore an increase in air pressure. Okay, so it decreases the volume at which the air is there, compresses the air, we get a higher air pressure. Okay, bish, bash, bosh. Right, let's move on and check the next thing. Ah, it's the end of the question. 4.5. The scientists repeated the investigation but made one change to the molasses. The scientists did not change the volume of molasses at the start of the investigation. So we kept the volume of molasses the same. The scientists observed the air pressure inside the tube increased by 160 kilopascals compared with 800 kilopascals in the first investigation. Okay. Suggest the change the scientists made to the molasses to cause the smaller increase in air pressure. Use the air pressure figures in a calculation to support your answer. So they've asked us to do a calculation. Well, we can see how much it only increased by 160 compared to 800. So first of all, let's find out how much less it increased. 800 over 160 is going to get us the first mark. So equals 5 times smaller increase. Right? So it increased by 800 the first time, only by 160 the second time. So it was 5 times smaller increase. So we're going to say the increase was 5 times smaller. There's our calculation for 1 mark. Why did it only increase 5 times smaller? Well, this means that the movement of the water from high to low was 5 times less water moved in. Why is that? That's because the sugar concentration was changed and the sugar concentration must have been decreased by 5 times. So we're going to say because the concentration of sugars in molasses was 5 times less or only 20%. So it was 20, they used the 20% of the original sugar solution. There we go. Boom. That one weren't a bad maths question. Okay, what we're having a look here is question 5. So 5.1, describe the appearance and the behavior of chromosomes. So the appearance, what do they look like and how are they behaving, how are they moving during the prophase and during the anaphase of mitosis. So what we might remember is, what does a chromosome actually look like during the prophase? Well, they form sister chromatids, 2 identical chromosomes joined together by a centromere. And so we call these chromatids. They're normally sister chromatids because they're identical to each other. And they're held together by a small little round thing called a centromere. A small little round thing, yeah, it's like my brain. Okay, so what happens during the prophase? Why is it that we can see the chromosomes? What happens to them during the prophase? Well, under an optical microscope, you can see the chromosomes or chromatids, you can call them either one, during the prophase. Okay, and that's because they shorten, they condense, and they become visible. So we can get 2 marks here for saying chromatids or chromosomes become visible because they shorten and condense. And if we want, we can even talk about how it's during the prophase that they attach to the spindle fibers. So the spindle fibers that are these thin fibers that run all across the cytoplasm of the cell and all of the different chromosomes attached to them. Now in the metaphase, they line up on the equator. So we don't want to mention that and get that mixed up. We just want to say they attach via their centromere to the spindle fibers, like I'm doing in this incredible level 4 GCSE art diagram. Boom. Enough whiffle waffle. That's going to get you 2 marks. I've kind of made 3 separate marking points there. You're only going to get a maximum of 2 on either of these. You've got to pick up 2 marks for each one. So although it's worth 3 marks, you can't get the 3. Right, anaphase. Think about the anaphase. Think of the A in anaphase is for away, right? The A anaphase is the away phase. What happens here is the sister chromatids, the centromere splits in 2 and the sister chromatids move apart to opposite poles of the cell, like I'm attempting to do here very badly. So there you go. You can see that those chromatids are separating and you can generally tell the direction from where the point is going anyway. Enough fiddling about with that. I'm not pulling that off very well. Let's type that answer out so you can get that down if you need to. So centromeres divide, or centromeres are torn apart, or centromeres split during the anaphase, and each of the sister chromatids goes to a different end of the cell. Now you can call them chromosomes when there's, I know they're chromatids, we say when they're together. There's still a chromosome when they're together. Both of them are a chromosome and they're called chromatids. And when they separate, they're really back to being called chromosomes. But the mark scheme will allow you to say chromatids or chromosomes. 5.2, a scientist used an optical microscope to determine the mitotic index in cells at different distances from the tip of onion roots. Figure 4 shows the results. Now, I can't show you figure 4 because of third-party copyright, but I have made my own one. Boom! Okay, so you can see my crusty figure 4 because this is not to be produced due to third-party copyright restrictions. So here is my impression of the image. It's not exactly the same because of copyright, but I've done my little impression of the image that gives you all the data. You kind of got a mitotic index up the side, and then you've got the distance from the root cap. So the further we get away from the root cap, what's happening to the mitotic index, there's a general relationship. It seems to be going down. Now, let's go on here. Complete the word equation used to determine each mitotic index in figure 4. So how do you calculate the mitotic index? You need to know this off by heart. If you don't know it, I suggest that you get out a revision card and you bust out this equation and learn it off by heart. So what you're doing is you're looking for the number of cells in mitosis. How will you know? Well, they'll be dividing when you look at them through the microscope. So you might be able to recognize the prophase, or you might see them lined up on the equator in the metaphase, the chromosomes, or you might see them moving away from each other in V shapes, which would be the anaphase, or you might see the cytoplasm kind of budding in and starting to pinch off into two, which would be the telophase. So the number of cells that are undergoing mitosis divided by the total number of cells in view is essentially the equation. So it should be the number of cells undergoing cell division over the total number of cells in the field of view. So maybe I need to put in the field of view, but we'd have to check the mark scheme on that one. But yeah, let's leave it at that. And that will get us a lovely fat mark. Right, I'm going to delete that now and let's go down. And of course, they could ask you something like, how long do these cells spend in the prophase? And you count up the number of cells in the prophase and divide it by the total. So it's kind of like a mitotic index for the prophase. You could then times that by 24 hours and work out how many hours in a day are spent in the prophase. Or you could look at the anaphase, count all the cells that are in the anaphase under the microscope, divide that by the total number of cells you can see under the microscope, and you would get like a 0 point number for the anaphase. You could multiply that by 24 hours and find out how many hours a day are spent in the anaphase of that particular sample of cells. Anywho, that's just a little bit extra because they sometimes ask you to do that. 5.3, the scientists used data from figure 4 to calculate the correlation coefficient R. So that's one of our statistical tests. That's the Spearman's rank we're talking about here. The scientists then used a statistical test to determine the probability P associated with the value of R. So um, our, our correlation coefficient R is equal to minus 0.98. When they look that up on that probability table, where you have N and you have degrees of freedom and whatnot, they found that they got this below this arrow to the left and then 0.05. What can you conclude about this result from that? Now, try to do this in your head, and then I'm going to go through it. So try to work out what they could conclude about that. And what they can conclude from this result, when we look at that graph, the graph was looking at a mitotic index and distance from the root cap. We're using Spearman's rank, or the correlation coefficient, to see if there is a correlation there. The null hypothesis would be there is no correlation between the mitotic index and the distance from the root cap. And what we would have found here is that actually there's a below 0.05 probability. So there is a less than 0.05 probability that the correlation in the results is down to chance. there is therefore a significant correlation in the results. So you reject your null hypothesis here would be that there is no correlation between mitotic index and distance from the root cap. What we're saying is actually there is a correlation, and there's a 95% probability or chance that this is significant. And there's only a 5% or a 0.0 or less than 0.05 or less than 5% that probability, I can't spell probability, there you go, probability that this is just down to chance. So actually, when you see the arrow to the left, and that's telling you the results are significant generally. When you see the arrow to the right or equals, that's telling you that the results are not significant because we Okay, enough of my whiffle waffle. Let's zing that there. I actually have a video on p-values right here. You can click on that and check that out if you like. All right, let's go down and see the last bit of this question. So the last bit of question 5, what can you conclude about the effect that the distance from the root tip has on the proportion of cells in different stages in the cell cycle? So let's get the image up and have a little waffle about this. It's 3 marks. Use the information. It tells us to use the information. What can we conclude about the effect of distance from the root tip on the proportion of cells in the different stages of the cell cycle? So let's check this out. Okay, so here's my crusty little graph, which mimics the one quite closely, but not exactly. So what can we see from this? Well, what's the general pattern? The general pattern is that what can we infer about this mitotic index? We've got a higher proportion of cells. Well, the cells have a higher mitotic index the closer they are to the tip. This is 0.5 millimeter from the tip, 1 millimeter, 1.5 and 2 millimeters from the tip. So what we can say is that over here, 0 is the tip. And what's going on? If we had to explain this, what's happening in the tip? What do we call the tissue in the tip of a root or shoot? We call it the meristem tissue. It's the cells that can divide and do differentiation. So they're doing tons of mitosis, and that's what we'd expect. We see there's a high mitotic index here. So what we can say for our first statement, for our first mark, is that close to the tip, more cells undergoing mitosis, right? Really simple. As you get close to the tip, there's more cells undergoing mitosis. We don't need to talk about meristem tissue and differentiation and whatnot at the tip of the root. We don't need to talk about that. So let's go, as we go down, the further away from the tip, what can we infer about these cells? If they're not in mitosis, this looks like only 20% are dividing. What other stage of the cell cycle must they be in? So have a think about it. What other stage of the cell cycle could you be in if you're not doing mitosis? So far from the tip, the cells are mainly in the in-wall interphase. Now, you could name any part of the interphase. You could say they're in the synthesis phase. They could be in G1 or G2. So far from the tip, the cells are mainly in the interphase. This is shown by the low mitotic index the further we go from the root tip. So that's going to get us like 2 marks. And then what can we infer about I should have really, actually, there shouldn't be a line here. On the actual picture, the line stopped here. So you can infer something from that. The fact that there are no cells dividing, well, the fact that that's like that it means that it's at 0, so it kind of rapidly drops off to 0. So there's no cells dividing. The mitotic index is 0 there. And we can also say that closer than this, so what we're going to say here, no cells are after 2 millimeters. After 2 millimeters, no cells. Oh, geez, Louise. Come on, Harry. Right. After 2 millimeters, no cells are in mitosis. All cells are in interphase. So once we're 2 millimeters away, now how can we tell that? Well, Unfortunately, I carried my graph on here, but it should have stopped there. If you've seen the real image, looks exactly like this, but the line stops there. So that after 2 millimeters, there is zero. The mitotic index is 0 after 2 millimeters, which means no cells are doing mitosis. And this would be your marks like that. All right, let's move on. Okay, let's go through this question. 6.1, give 3 structural differences between mRNA and a tRNA molecule. It's worth 3 marks. So why don't you pause the video, grab a pen, grab some paper and scribble down what are the differences. Okay, I'm going to go through the answers now and you can see how many you got right. There's about 5 or 6 different differences, but we'll go through them. So if you imagine DNA over here in pink, mRNA is a single short strand of a section of DNA. It's basically a copy of a gene, right? And what is tRNA? Well, tRNA is the carrier molecule that is going to deliver an amino acid in transcription to the ribosome. Now, tRNA has a different shape. How would you compare that blue drawing of mRNA to the shape of tRNA? How would you compare that? And what is attached to a tRNA molecule that isn't attached to an mRNA? So let's get some of these differences down. So first of all, dealing with the shape, if we look at the blue mRNA on the left, it's linear, meaning it's just basically a line, a straight piece. They're not joined at the ends. But if we look at the tRNA, it's clover shaped. It's basically a loop of DNA, isn't it? A circular piece of DNA joint at the ends, the green one there. Also, mRNA has codons, which is the nucleotide sequence, which is kind of opposite, but joined by complementary base pairing to the anticodons on the tRNA. So I'd be like mRNA has codons, tRNA has anticodons. And now the tRNA has an amino acid binding site, whereas the mRNA has no amino acids attached. Okay, let's go over a couple of other ones. So if we look internally within the tRNA, these are all nucleotides and there's intermolecular forces between them. That's a really rubbish drawing. Let me just rub that out. Excuse them. What? Okay, let's redo that. Okay, so here is our clover bit of tRNA. Now, what I'm trying to show you is that it will have like guanine on the inside and cytosine opposite, and they'll hydrogen bond to each other, hence why it's in a loop or a clover-shaped loop. Whereas the mRNA is linear because there is no complementary base pairing with another nucleotide. So there's no hydrogen bonds. Okay, looking at this next question, 6.2, table 3 shows mRNA codons and the amino acid coded by each codon. Okay. Figure 5 shows the mRNA base sequence produced when part of a gene coding for an enzyme is transcribed. So what we're going to do is we're going to go through, it says use table 3 to give the amino acids coded by the mRNA base sequence in figure 5. So what we're going to go through here is try and work out which amino acid is coded for by each of these 3 triplet codons on the mRNA. So we've got UUU, CGG, and GCG. So we're really learning here how to read this kind of table 3, which is a very common table to be able to read when you're dealing with DNA. Okay, so let's have a look. We've got UUU, CGG, and GCG, and we need to work out what these are and stick them in these blank boxes. Let's do it now, folks. Right, so looking at the first one, you're going to circle the first U. So it means that the amino acid is found in this row. And then it's also, we look at the second base, which is also U. So it has to be in this column. So it's got to be PHE or LEU. Now, we can see the U is there, and we're going to go across, draw a line across. It has to be PHE because that third base is in line with that. So our first amino acid is PHE. We're now going to look at the second one is CGG, and that means because it's in this row where I've circled in green, it's one of these 5 amino acids. It has to be in that column, and it doesn't matter which one it was in that column. They would have all been arginine, ARG. Okay, now let's look at the third one. I'm going to pick another color. So we've got G first of all in brown here, then C, and that tells me it's in this row. It has to be in that column. It's going to have to be alanine. We don't even need to look at the last one. So it could have been G, could have been U, could have been whatever it was. the code GCG codes for alanine, but GCA or GCC would code for alanine as well. And there you go. It's a lot of work for 1 mark. Stingy AQA. 6.3. A mutation occurred in part of the gene transcribed in question 6.2. Figure 6 shows the mRNA base sequence produced when the identical part in the mutated gene is transcribed. So we've got UUU, AGG, GCG. The amino acid coded by this mRNA base sequence forms part of the enzyme's active site. Use all the information in the question to name the type of mutation that occurred to produce the mutated gene, give the change in the DNA caused by the mutation, explain the effect this mutation will have on the function of the enzyme. It's worth 4 marks, and they've spaced it out nicely for us like this. So what we need to do is zing back up and see what are the differences. And if we have a little look here, we've got UUU and UUU, so there's been no change, no mutation there. And we've got GCG and GCG, so no mutation here. However, in the third one, we got CGG, and this has turned to AGG. So there has been a swapping of one of the bases. C has swapped for A. So that is what has happened. And we call this a substitution mutation. You can call it a silent mutation as well, as we'll come on to with our explanation. Now, what is the change in the DNA? This is the bit I'm going to forget. Was it C got changed to A? I think it was. So on the second codon, the middle codon, a C was swapped to become an A. So the C base was swapped for an A. Let me just double bubble check. Yes, that's right. Now, what is our explanation of the effect on the function of the enzyme? So we're going to have a little look up here, and what we can see with CGG, that is arginine, right? Now we have gone to A. Oh God, I've done that wrong. Not ACG, you idiot. We're talking AGG. Whoopsie. Hold on. Let me delete this. Okay, what I'm trying to do is show you the comparison so we can actually work out what's happened. Has this changed the amino acid, or is it for the same amino acid? So you'll see here, instead of being CGG, it went to AGG. Let's do that again. A's up there, G's there. I'm doing it in pink. It also codes for arginine, right? So it doesn't matter the fact that this C swapped for an A. They both code for arginine. There we go. Finally got to the bottom of it. Sorry about that, folks. A little bit of a hiccup there. So it is a silent mutation. And what this means is, as DNA is degenerate, meaning that more than one codon there's more than one codon coding for an amino acid. Even though the C has been substituted for an A, they both still code for the amino acid arginine, both of these triplets. What does this mean in relation to the enzyme? Well, this means that the enzyme will have the exact same primary structure, excuse my spelling. What is the primary structure? That is the structure that is going to inform the type of bonding that we're going to have, because it gives you the type of R groups, and that will decide what type of ionic, covalent, hydrophilic, hydrophobic interactions or bonds you form in the tertiary structure. So it means that even though it's been mutated, it has the same tertiary structure, the same 3D shape. The enzyme's active site will still be exactly the same, and it will still be functional, and it will still form an enzyme substrate complex. So the enzyme will be fully working even though it has had a mutation. This is called a substitution. And now for the second mark here, it says the change in the DNA. Well, the change in the mRNA was C to A, so cytosine was swapped to adenine, and that means that in the complementary DNA template that coded for the mRNA, the swap was guanine to thymine. So they actually need you to say that G was swapped for T in the DNA, even though we're given here the mRNA C for A. Well, C would be complementary to guanine on the DNA. So normally guanine would produce the cytosine in the mRNA, but the guanine was changed to thymine. So when the mRNA was coded for, it was adenine instead of cytosine. Complex? Well, not that bad. All right, let's move on to the next question.
You still here? It's over.
Okay, 7.1. A student investigated the activity of the enzyme polyphenol oxidase, PPO, in apple tissue. When apple tissue is exposed to air, PPO catalyzes a reaction between colorless phenol compounds in apple tissue and oxygen. Figure 7 shows the reaction. Phenol compounds plus oxygen in the presence of PPO turn to a brown pigment. We all know apples go brown when you cut them, right? The student measured the time taken for the brown pigment to appear in two apple varieties, D and E. Here's their method. Number one, they cut a 1 centimeter cube of apple tissue from variety D. They put the cube on a plate and leave the plate at 30 degrees. Measure the time for the brown pigment to appear. Repeat steps 1 to 3 two more times. Repeat steps 1 to 4 with apple tissue from variety E. The student obtained the results shown below but did not record the data in a results table. Variety D equals 15 minutes 50 seconds, 18 minutes, and 14 minutes 30 seconds. So we got 3 times there. Variety E was 6 minutes 30 seconds, 8 minutes, and 7 minutes. In the box labeled Figure 8, design a suitable results table, enter the student's results into the table, calculate the mean results, and include these in the table. Use 1 decimal place for both mean results. So what we need to do here is zing down and we're going to find, a results table. And we need to scribble our results table there using all the information up there. So I'm going to do my table here on the side. And essentially, oh my gosh, pink. Yes. Right. What we're doing, generally the rules for the results table, you might want to pause the video and have a go, but the general rules are independent variable, what you change, goes in the first column, and then your dependent variable in the second column. So why don't you have a go? And then I'll go through and explain the answers. Okay, let's go. Independent variable is always in the first column. And what did they change? Let's scan read. Okay, they didn't change the PPO, the enzyme catalyst, the phenol compounds. They measured the brown pigment. Two apple varieties, D and E. So we've got our first heading should be apple varieties. And we've got 2 of them. We've got D and we've got E. So what you change, which is the independent variable, is always, oh my good gosh, is always going to be in the first column. So apple varieties D and E. Now what we want to do is look at the independent variable, the dependent variable. Okay, what we're going to do here, we are going to add a third column. Oh my gosh. For our mean. So what we're going to do, apple varieties, and then we want times. Now, we've got a little bit of a problem here. We have to make a choice. When we look, your dependent variable is what you measured, and they measured the time. Okay, time to go brown, I should call that actually, rather than just times. Let's give it a little bit more nuance. Say time to go brown. I mean, that's still a pretty crusty name, but there we go. Time for the brown pigment to appear would have been a better thing. Time for the brown pigment to appear would have been a better title, but I'm just going to leave it time to go brown. Now, we have an issue. They've mixed the time units. Look, we've got 15 minutes, 50 seconds. That's like saying meters and centimeters. They're 2 different units. So you need to either convert these to minutes or to seconds. Now, it's a lot harder to convert these to minutes. I'll give you an example. 14 minutes and 30 seconds. It's quite easy to convert to minutes, right? It's 14.5 minutes because that's half a minute. But how do you convert 15 minutes and 50 seconds to minutes? That's a little bit harder. That's 15.8 minutes, but that's going to be harder for you to do the conversion. I would just convert these all to seconds, and then that's going to make it a lot easier. So I'm going to do my time to go brown, and I should write the words in and then put seconds, but I'm running out of room, so I'm just putting S in brackets. So time to go brown in seconds, and I'm going to do 15 minutes and 50 seconds. So I just want to do 15 times 60, and that equals 950 seconds. So I'm going to have 950. I don't need to write the unit because it's in my heading, and that's the beauty of putting your unit in the heading. Then I want to do 18 times 60, and that's going to give me 1080 seconds. Again, I don't need to write the unit. It's in the heading if you put it properly. And then I want to do 14 times 60, and that equals 840, but I need to plus 30 because it was 14 minutes and 30. and I've got 870 seconds. So don't mix your units and do minutes and seconds. Either convert these to minutes, which is a little bit harder, or convert them to seconds, which is nice and easy. Okay, now I need to calculate the mean, and this is going to be my mean time. Um, and really what I should have done before is write in and then put S in brackets in seconds, mean time in seconds. I prefer to write that word IN just to make it really clear. So now what I'm going to do is add those all together, divide by 3, and then they did say to one decimal place. So let's do that as well. Ba, 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 ba. Um, I hope this is right. Divided by 3. So I got two for this first mean time. I got 2,900 divided by 3 gave me like 966 point 0.6 recurring. Now I'm going to round that to 966 and I'm going to go zero point 0.7 as my mean time. Now I'm going to do E. Okay, so for E, the results are 6 minutes and 30 seconds. So I'm going to go 6 times 60 is going to be 360. And I'm going to plus 30 because plus 30 seconds. So I'm going to get 390 for my first lot. Now, the second time they did it with the apple, it took 8 minutes to go brown. So I'm going to go 8 times 60. And that's going to give me, God, my eyesight's bad, 480. I could easily make a mistake here. And then I'm going to do 7 times 60, and that is 420. So now I've got all of that. Again, notice I didn't have to write the units. It's in the heading. That's what they want you to do. So I'm now going to go 420 plus 480 gives me 900 plus 390 gives me 1,290. So this equals 1,290 when I add all three of those up. I'm going to divide by 3. And then remember, we need to round to one decimal place. But this time I get 430.0. Yep, I got 430. And that is my lovely jubbly answers. So just to double check, make sure you might have made a mistake here. You might have gone 430 and not gone to zero point 0. They were really clear. They wanted it down here, you'll see, to one decimal place for both mean results. Look, they were really clear for both. I would have thought they put that in bold black just to give us a hint, but they want both, even though this one rounds to a whole number, 430, they want it 0point 0 there. Otherwise, you're going to lose a mark. They would want all of these time units in converted either into minutes or into seconds. I've done them into seconds. And then they want the independent variable, which is what you change at the start in the first column. They weren't really too bothered whether you put the mean time first and then the time. But originally I thought, well, I won't include the rough times. And then I read that again and I realized, hey, the reason why they've given all these weird like minutes and seconds like that and then whole minutes is because they want us to carry out that calculation. So we get 3 marks, one for the independent variables D and E in the correct place, one for all the correct times, which is the hardest bit, converted to the correct units, whichever one you chose, and then one for both the mean times, both to one decimal place, including 430 to 0.0. Okay, let's move on. I'm ticking the Lumin worksheet. Okay, so that would have all been in that space there. Now it says 7.2, suggest and explain why the results for variety D are different from the results for variety E. So what we have to look at, what is it they didn't control that might mean that D, because we'll see E had a much smaller mean time. It went brown a lot quicker. So what is it that could be different? Well, let's check their method out. Difficult one, but it's something they haven't controlled about this method. Let's read right from the top. Let's read. A student investigated the activity of the enzyme polyphenol oxidase. There's nothing in the method about the enzyme. Like, what concentration did they use? What volume did they add? Was it the same? They haven't told us whether they controlled that. So maybe they used a higher concentration of polyphenol oxidase with variety E, hence why it only took 430 seconds as opposed to like 900 seconds to go brown. Or maybe they used a larger volume and yeah, so they haven't mentioned the enzyme. So I would go with that. We're going to have a look. Oh, phenol compounds plus oxygen. Maybe one variety had a higher conc of phenol compounds in it just by the nature of its species or variety. So higher phenol content or concentration, right? And that is what is causing that brown color when it reacts with oxygen. Cool. They have kept the surface area the same as they're both 1 centimeter cubed, so we can assume they've kept the surface area the same. There are probably some people who wrote that, but yeah. And so what I would go with here is it says, and look, keywords, folks, keywords, suggest and explain. That means the word explain means we need to bust out the science. So what we're going to do here is say different concentration. Let's say I'm going to give variety E had a higher concentration of PPO enzyme, meaning more enzyme substrate complexes could form. So faster rate of producing the brown pigment or the brown product, right? So it's doing it's catalyzing that reaction faster because variety E, when they use variety E, they use the higher concentration of the PPO enzyme. Alternatively, we could say with this is just a separate answer, with variety E, it may have had a higher concentration or more higher amount or volume of phenol compounds in the apple. Again, this means more enzyme substrate complexes. This time the phenol is not the enzyme, the phenol is the substrate. So more enzyme as well as substrate complexes form. So higher rate of catalyzing. Yeah, whatever. Okay, and that's going to get us the 2 marks. So it's clear here they wanted to suggest and explain. So suggest is higher concentration of enzyme or different amount of enzyme, a bigger volume of enzyme. When they carried it out, they forgot to control that in the method. That was a method mistake. The other one isn't really a mistake of the method. It's just that variety E and D may naturally have different amounts of phenol within their cells. Or produce different amounts of phenol. So they're going to naturally react to different rates because of that. And the explanation is mentioning enzyme substrate complexes, basically. So bish, bosh. Okay, let's zing. 7.3. The student repeated the investigation but made one change to the method used to prepare the apple tissue. The student then observed shorter times for the brown pigment to appear in both apple varieties. Suggest the change the student made to the method of preparing the apple tissue. So there's something they've done that means it goes brown quicker. Explain why the brown pigment appeared in a shorter time. So again, this is the same as the last question, really. Just to repeat, we've got to suggest and then explain why the brown pigment appeared in a short time. Do not suggest using a different volume or mass of apple. So we can't really suggest like, oh, you used more apple. So we can't say there was more phenol compounds. Why can't we say more phenol compounds? Because you can't control that. That's produced by the cells of the apple, right? That's not controlled by the person. We're not injecting the apple with phenol compounds. So change to the method I'm going to go. You go up to the method and you look, what did they control? And you might remember they controlled the surface area, right? They did 1 centimeter cubes. So we could change that. Or they did it at 30 degrees on an agar plate. So we could change to 30 degrees. So I would either change one of the two things they controlled, and they controlled temperature or surface area. So I'm going to say change to the method, higher temperature, and that's going to get you a mark. Or we want to say smaller cubes, larger surface area. So we're going to go, or you could say, or smaller cubes, larger surface area. What I mean by that is same mass, because they said you keep the mass the same, but you're using smaller cubes, so it will have a larger surface area. Now, the explanation for the higher temperature, why does that react faster? What we want to say is more kinetic energy, more successful frequent collisions between enzyme active site and substrate, resulting in more substrate enzyme complexes forming per second. I'm trying to give it a time frame. So essentially, more kinetic energy, more successful collisions between the active site and the substrate. That is going to get me those 2 marks, and that goes with the temperature one. Now, if you went down the surface area route, what we want to do is slightly different answer, but it's pretty much, you're just changing the kinetic energy. So you're going to say more substrate available. You've made more surface available, so more substrate, more substrate available for collision with active, sorry, site of, I've got fat fingers, of the enzymes. So more substrate available, more surface available for collision with the active site of the enzyme. And again, more, I'm just going to put ES complex, guys. I can't be bothered to type that out. More ES complex form. There we go. And so that is your 3 marks there. Surface area, more substrate available for collisions with the active site of the enzyme. So more enzyme substrate complexes form. And there we go. That's 3 fat bad boy marks. Now let's go down to 7.4. And this is the last one of the question. Let's zoom in. It's a little multiple choice MCQ. The student wants to change the procedure to obtain a measure of PPO activity, either in terms of how much substrate is used or how much product is produced. So they want to change the method, the procedure, same thing, to actually get a quantitative measure rather than just looking at the brown pigment of PPO activity, either in terms of how much substrate is used or how much product is produced. Which change in the procedure will provide a successful measure of PPO activity for the student? Measure the increase in oxygen concentration in the air around the cubes of apple tissue on the plate. Let's go look at the equation. We're moving on up. Okay, check this reaction. Phenol compounds react with oxygen in the air. Is the air oxygen going to increase or decrease? Obviously, the oxygen in the air is going to decrease because it's joining with the phenol to make this brown pigment, some sort of iron oxide. So when we look at it, actually, we would expect the oxygen in the air to decrease. So measuring an increase in oxygen in the air is going to be the wrong answer. Yeah, look, there's going to be no increase in oxygen concentration in the air. It's going to decrease. So that one's wrong. Measure the intensity of brown color by comparing apple tissue with a color chart showing a range of apple tissues of known pigment concentration. That one sounds all right, like they've done some sort of calibration curve, like different browns correspond to different concentrations of that brown pigment, that iron oxide or whatever it is. Measure the intensity of the color produced from the brown apple tissue in a Biuret's test. Now, what does the Biuret's test for, guys? The Biuret's test tests for protein. And it goes purple or pink. Does it go brown? No. So that would be really crusty to use that because the Biuret's doesn't go brown. So you can't really compare it to the apple tissue. Measure the percentage of light transmitted through a cube of brown apple tissue using a colorimeter. Now we might say, oh, hold on a minute, this one sounds pretty good. How much light can get through an apple? Well, can light get through an apple? The answer is no. No light's going to get through an apple, whether it's brown or not brown. So using a colorimeter is pretty rubbish on that. If we wanted to use a colorimeter, we'd have to make like a solution or something where the light is going to be able to get through. So that's going to be a bit complex. So we're not going to do that. This one sounds perfect. Measure the intensity of the brown color of the apple. Compare it to a range of known brown colors where we know, oh, that one's got this concentration of iron oxide or whatever it is that makes it brown. Awesomeness. Right, let's put a tick there. And let's zing that there and get rid of those little question marks. And that's the end of that question. Don't forget to, hit the like and all that jazz. I'll catch you on the next one.
You're still here? It's over.
Give 2 structural features of an aorta wall and explain how they are related to the function of the aorta. 2 marks. Don't forget it's an artery. It's the artery that pumps the blood from the heart to the body. Here's the structure. Pause the video. Have a go. Okay, so we've got muscle and elastic. We've got a little lumen in the middle. We've got an endothelium layer, single layer of cells on the inside. We've got the protein coat on the outside. So you can name any of these structures. Muscle and elastic fibers are really 2 different structures. So let's go through them. Endothelium is the inner layer of cells in contact with the lumen. They're smooth, so they reduce the friction of the blood as it goes past, reduce the chance of you having a blood clot or anything like that. Muscles can contract and relax. You won't get marks for that. But the muscles allow the walls to withstand pressure. They basically give strength to the walls and allow them to withstand that high pressure. Elastic fibers have a slightly different drop. They stretch and then recoil, they spring, which enables the blood pressure to be maintained at a constant level. And the tough protein outer layer prevents cracks or prevents the artery from bursting due to the high blood pressure, because this has all come from the powerful left ventricle muscle contraction. Okay, that's 8.1. Let's zing, zing, zing. 8.2. Small tears may occur in layers of tissue in the aorta wall. These tears weaken the wall without bursting the aorta. Scientists measured the aorta diameter D in a large population of people over 60 years of age, and they calculated the risk of the aorta wall developing a tear. Table 4 shows the results. We have the aorta diameter, we have the number of people in the population who have this diameter, and we have the risk of an aorta wall developing a tear. So blood may push through the tears in the aorta wall. This produces a balloon-like swelling called an aneurysm and increases the aorta diameter. Aneurysms can cause the aorta to burst. Using all the information, what can you conclude about the aorta diameter and the risk of developing an aneurysm? And it's 3 marks. So conclude. Listen, when you see the word conclude, it means pure facts. Don't go explaining, talking about high blood pressure and stuff like that. You don't need to do that. Just say what you can see from the numbers. Uh, any proper good points you can see in the numbers about the aorta diameter and the risk of developing an aneurysm. So straight away, we can see the general trend is that as the aorta diameter increases, what happens to the risk of developing a tear? So we want to say, as aorta diameter increases, risk of developing a tear increases. And what is the tear linked to? If you have a tear, it means you're more likely to have an aneurysm, right? Because you get the tear and then you get the balloon-like thing, and you're more likely to get the aneurysm. So what you need to do here from your conclusion is link the fact that the risk of developing a tear means more tears means more likely to have an aneurysm as well. And that's going to get you that first mark. However, what hasn't been shown from the data is that if you get a tear, you definitely get an aneurysm. That's not true, is it? It doesn't. We don't have a column here saying risk of aorta developing a tear and then tears develop into the percentage of tears that develop into aneurysms. We don't know that. So we can put that as a statement as well. We don't know how many tears become aneurysms, or not all tears will become an aneurysm. They don't necessarily all lead to an aneurysm. So that's a point worth making that we can conclude from this as well. And there's loads of things you can conclude. We can say that it's a very, very small chance, 0.06, for the majority of the population. They have a small diameter. So most of the population have a diameter below 3.5 centimeters, and so have a small chance of developing a tear. We'd say the highest, only a tiny number of the population, 7 people, have a diameter of the aorta above 4.5, and they have a very high risk of developing a tear and therefore an aneurysm. So you're just kind of stating facts about any of these ranges of aorta diameter. You could do any of these, really. You're just talking about that most people have small aortas and don't develop tears. Right, 8.3. A scientist investigated changes in a diseased heart and changes in a healthy heart. For each heart, the scientist measured and obtained a value for the mean volume just in the ventricle just before it contracts, called BVB, so the whole blood in the ventricle, the mean ejection fraction, basically the stroke volume, how much blood is coming out, the proportion of blood pumped out of a full ventricle in one heartbeat, that's basically the stroke volume. The EF is calculated using this formula. EF equals the blood that gets pumped out of the heart over the total amount of blood in the ventricle. So imagine this is the blood here I'm drawing in this crusty little drawing flying out of this could be the left or right ventricle, but let's say it's the left ventricle, right? So and this is the blood coming out. Now the blood coming out is the stroke volume, or the blood pumped out during one heartbeat. So blood, no, sorry, I can't spell. Blood out divided by BVB equals the ejection fraction, the fraction of total fraction of the blood. And we've got a little table here that shows the results, table 5. We've got the mean blood volume for a diseased heart and a healthy heart. So a healthy heart has a bigger amount of blood stored in the ventricle. And then we've got the mean ejection fraction, and we can see the ejection fraction is bigger for the healthy heart as well. Now, what's our question? Using table 5, a student calculated the percentage change in the stroke volume of the diseased heart compared with the stroke volume of a healthy heart is minus 30%. Okay, the student's answer is wrong because the final step of the calculation was performed incorrectly. Use the equation in table 5 to calculate the correct percentage change in the stroke volume of the diseased heart compared with the stroke volume of the healthy heart. Identify the mathematical error in the final step of the student's calculation. So we've got to find out what they did wrong. But first of all, we need to actually calculate the percentage change in stroke volume. So the stroke volume here, remember, is the blood out. So they're basically asking us, what's the percentage change in the blood out? Now, the table doesn't tell us the blood out, but we have the proportion of the blood out, the ejection fraction, EF, and we have the total amount of blood in the heart. So we can work the amount of blood that went out. When I say blood out, of course, I mean SV, stroke volume, which I'm circling here. So what we're going to do is rearrange this equation. Blood out divided by total blood volume equals ejection fraction. So if I times ejection fraction by BVB, I'm going to get the stroke volume or the blood out. Okay, so that's going to give me the stroke volume. So all I've got to do is 100 times 0.45 for the diseased heart, and the amount of blood that leaves the heart with one beat is 45 centimeter cubed for a diseased heart. Let's check out how much blood leaves a healthy heart with one beat, and it's 120 times 0.58. And if you do 120 times 0.58, you're going to get something like 69.6 centimeter cubed. So there we go. Now what we want to do is we've calculated the stroke volumes, so let's now work out the percentage change. Do you remember the calculation for percentage change? You should know this equation off by heart 100% in your A-level biology in paper 1, 2, or 3. This is coming up. Okay, you're going to get a percentage change question. They almost have to ask it every year. So what is the percentage change? Well, the percentage change is the difference between your two sets of data divided by the original in the timeline, the first set of data. So here we're going to do the difference between the diseased heart and the healthy heart over the healthy heart. Okay, so technically here we're going to do 69.6 take away 45, and that is going to get us the difference in the stroke volume is 24.6 centimeter cubed. We're now going to divide this by the original, which is 69.6 centimeter cubed, and we're going to times that by 100, and that's going to give us around about 35%. So about 5% different than the student's calculation. It was 35 point, let's whack that in the calc. Okay, so I got 35.34. You might want to round that to 35.3. Yeah, 35.3% would probably be better to 1 dp. And there we go. What's the error? Well, they've obviously just not rounded it correctly. They've done incorrect rounding. And where did they round incorrectly? They might have rounded 45 to 50 and 69.6 to 70. Smash that in the calculator in your percentage change, and you get like 28%, and then they've rounded up to 30%. So they may well have done that by incorrectly rounding the stroke volumes or incorrectly rounding the final percentage. So they've messed up in some way. Okay, let's move on to question 9 and get this paper done. Question 9. Courtship behavior in the frog species Xenopus laevis involves male frogs calling to attract calling to attract sexually active females. These are advertisement calls. You want sexy time? Or start and continue mating. These are mating calls. I won't do one of those. Signal when a male is not sexually active. These are rasping calls. Got a headache tonight. That's the rasping call. Scientists investigated frog courtship behavior by feeding a population of sexually active male frogs a diet containing the hormone EE2. The scientists also fed a separate control population of sexually active male frogs a diet without EE2. So they used two lots of sexually active male frogs. ones with the hormone EE2 and ones without. They determined the percentage of males making advertisement calls or rasping calls in each population. So they did the advertisement, the top one, and the third one, the rasping call, but they didn't do the mating calls. Okay. 9.1, there were 800 males in the control population. Each male made one type of call. Use this information in Table 6 to calculate the number of males making mating calls in the control population. So pause the video, have a go at this calculation. Right, I'm going to go through it. So let's have a little look. Now, what you can see from this, they want us to calculate the number of males making the mating calls. Well, we've got the advertisement data and rasping, but we don't have the data on mating. So what were we looking at? The control population. Which one of these is the control? Well, it says right there, the one without the hormone is the control. So we know that 97% were doing advertising and rasping was 0.5. So we can work out this one by doing 100%. And we want to take away 97 and take away 0.5, and that equals 2.5%. So there's 2.5% that are doing the old mating call. Okay. Uh, we know the total population is 800. So how do we work out 2.5 of 800? You do 2.5 divided by 100. What's that? 0.0. Oh gosh, I'm not even going to attempt. My maths is so bad. 2.5, just smash that in. I was going to say 0.025, but it's I'm probably out. So 2.5 I'm used to 2.5 divided by 100 equals 0.025, yeah, times 800, 20. So there's 20 little froggies that are either at the start of mating or during mating making those calls. Okay, that's going to get us a little tick. One of those beautiful maths questions. Okay, let's zing down. Suggest one change the scientists could make to both frog populations to increase the number of mating calls. So these are male frogs. What could they do to the frog population that will make them call out to do basically more mating calls, not advertisement calls? Advertisement calls are when you're saying, come over here, let's get it on. And rasping calls are the ones where you say, I'm not interested, I'm not interested. But the ones where you're actually mating continue to mate. So making the noises during the mating, how could we increase the number of that? Well, we could add females to the group. Okay. So we could add female frogs. Then they're going to be able to start mating and they'll make those mating calls, meaning that they are in the process of mating. Okay. Um, let's zing down. This is quite a short question. So let's zing down to the last bit of the question. 9.3. The scientists also investigate the effect on female frog courtship behavior of feeding EE2 to male frogs. So they're not giving the hormone to the female frogs. They're just seeing how did it affect the females. Table 7 shows the result. So the males fed EE2. Uh, the amount of time in seconds the females spent doing the do with them was 8 seconds. Boy, that's quick. Whereas the males who weren't on the hormones, the control, they spent double the time, 16 seconds, the females spent mating with them. So they spend half as much time with the EE2. EE2 is contained in human contraceptive pills. Some EE2 is released in human urine and collects in the sewage. So when we go for a wee-wee, that hormone is in the toilet. Untreated sewage pollutes the water in frog habitats, so ponds, lakes, rivers. So Justin explained the effect of EE2 pollution in frog habitats. What will it have on frog breeding? We have to use the information from table 6 and 7 in our answer. It's 4 marks. 1 mark for the effect on frog breeding, 3 marks for the explanation. Make sure you make 2 points from one of the tables and 1 from the other. Okay, pause the video, have a go. I'm going to go through the answer now. So obviously it looks to us that EE2 is going to decrease. What is it going to decrease? Breeding. So it's going to decrease the amount of breeding. How do we know that? Well, the males fed EE2, the females spent less time in courtship. So we can say because females will spend, less time in courtship. Um, Only like 8 seconds compared to 16 seconds. So they're going to spend half as much time in courtship because of the E2. So that's going to get us a mark, and that is data from table 7. We now need to go up and take data from table 16. So let's go upstairs and have a little look. Ba-da-ba-da-bum. We can make some points here as well. So just, ooh, where's my answer? Bringing it back. Bring it back. Bring it back. Bring it back. Right, I'm right. I'm going to leave my answer here because then I've got this table here. Okay, guys. So this is part of the 4 marker down below, but I can't bother to scroll down. So males fed EE2. So what we're going to get is less advertisement calls. So also less advertisement calls. So and more rasping calls, meaning less frogs are sexually active. So there's less frogs that are sexually active, and there's less advertisement calls. Which what does that mean? So less females attracted. If you're not kind of asking the females out, they're not going to go with you. And more rasping calls to say, not interested, I'm staying in and playing on the Xbox. So yeah, any of those. And I think there's another one we could go for here. There's up to 5, even though it's a maximum of 4 marks. Males favorite, we've got advertisement calls, rasping calls, and then yeah, all that will do, won't it? 2 from there, 1 from the data table down below, and then EE2 decreases the breeding. Let's leave it at that, and that's the end of that question.
You're still here? It's over.
Okay, we're on to question 10, the content questions at the end of paper 1. Some people recommend to do these when you start your exam right at the start and get these solid content marks out of the way. Okay, let's get on. Describe and explain how you would use cell fractionation and ultracentrifugation to obtain a sample of nuclei from muscle tissue. So here's some muscle tissue, right? I'm just going to sketch it out. By the way, why don't you grab a pen and paper, pause the video, have a go at this on a scrap of paper, see how many bullet points you can get, and then I'm going to go through it. Okay, here we go. So first of all, you want to homogenize the tissue. Now, what does this mean? This means blend it, break it up, and this is going to release cells from the connective tissue, like the ligaments, tendons, elastic tissue that's holding the cells together, okay? And also the homogenization or homogenation, no, not homogenation, homogenization or whatever, is going to break open the cell membranes and release the organelles. So we've now got our mitochondria, ribosomes, Golgi body, whatever, all released. Okay, from the muscle tissue. Now, how do we separate these? Well, for a start, we need to use a solution that's buffered. What does buffered mean? It means that the pH doesn't change much. And that's important because changes in pH can denature proteins, which will change the 3D structure of proteins. And yeah, you know about denaturing proteins. So maintain the pH with a buffered solution. You should use a cold solution. The reason it's cold, less kinetic energy, you're going to reduce enzyme activity. So you're going to stop enzymes breaking stuff down, like your proteases and that type of stuff. Okay, use an isotonic solution. Isotonic means that the cytoplasm of the cells and the cytoplasm of the cells is the same water potential as the solution you're using for your cell fractionation and centrifugation. And this prevents an osmotic effect taking place. It prevents the net movement of water into or out of organelles that could cause things like your mitochondria to explode or to shrivel up. So we need to use an isotonic solution. And that's going to get us a whole bunch of marks. We've basically described how to do cell fractionation. That is basically cell fractionation. We're now on to ultra centrifugation. Now, ultra means you're dealing with really small stuff, and the organelles are very small. And centrifugation is that machine that spins, and you can change the RPMs, how quickly it actually spins. And if we spin the machine at low speeds, the nuclei, because they're the heaviest organelle, will float down and settle out as a solid pellet. And as I don't require any of the other organelles like mitochondria, Golgi apparatus, etc., I can throw away the supernatant. The supernatant is the leftover solution which has the other organelles in. If I wanted to get them out, I would spin the centrifuge at different speeds and they would come out at higher speeds. Okay, um, that is going to get me 6 bad boy marks. Yeah, don't forget that I forgot to say you should filter after homogenizing and blending the tissue to release the cells and release the organelles from the cells. You should filter to remove debris. Now, moving on to the next part of the question, we've got 10.2. This one's worth 5 marks. Describe the role of organelles in the production and release of enzymes by animal cells. Do not include details of transcription. However, what they haven't said here, if they've said don't talk about transcription, what could we talk about? Well, we could still talk about translation, right? Why don't you grab a pen, grab some scrap paper, have a go at this, try to make 5 bullet points about how proteins are made in a cell, i.e. enzymes are proteins. So how are proteins made in a cell, and then how are they released to the outside of the cell? All right, I'm going to go through the answer. So you got DNA in the nucleus, so you can if you want, but don't worry if you miss this bit because you can still get all the marks. DNA codes for the primary structure of a protein. You're not really going into details of transcription. You're just saying that the nuclei or the nucleus plays a role. So, what next? Well, once that bit of mRNA, of course, leaves the nucleus via a nuclear pore, we're not putting that in, that's details of transcription. We want to talk about the translation section. So, amino acids are assembled where? Well, that can either be on the rough endoplasmic reticulum. I've used abbreviation. In your exam, don't use abbreviation unless you have specifically written the name first. So, write rough endoplasmic reticulum first, then if you're referring to it again, use RER. Now, proteins can be translated on a free ribosome in a cytoplasm or on the RER. That will be the same marking point. Okay, so a mark there for talking about translation where it occurs. You also get processing of the protein. This means it develops its secondary structure, you know, the alpha coil or the beta pleated sheet, etc. And that is inside the rough endoplasmic reticulum. It can be folded. Then it needs to go and get packaged. So it's going to travel via a vesicle to a Golgi apparatus. But before that, we could talk about the fact that assimilation of amino acids on the ribosome in translation requires ATP, and which organelle makes ATP or releases or produces, sorry, actually makes ATP? That is the mitochondria. You won't get the mark for saying energy here. You need to say ATP is required from the mitochondria for protein synthesis. Now, as I mentioned earlier, the Golgi apparatus, that is where the protein is packaged and modified. It could become a glycoprotein there, have a glucose molecule jammed on it, so it becomes a receptor protein. So it's going to be packaged and modified there. How does it get there? It travels there in a vesicle. I could have put that. I didn't because I'm going to talk about vesicles after the Golgi apparatus. But you could talk about vesicles in 2 places here. Proteins are transported to the cell membrane inside a vesicle is going to be a mark. And then I want to say, might not be on the mark scheme. Often it says ignore, but the vesicle fuses with the cell membrane because a vesicle has a layer of phospholipids around the outside. So it can fuse with the cell membrane, become one with it, and release its contents outside the cell by exocytosis. And if you go through all of this, there may be some bits that they're going to ignore, or we may have said things that are on the same marking point, but we're easily going to pick up 5 marks. And here we go. That's how to do that question. Don't be afraid to kind of bullet point your answer without the bullet points. Have like invisible bullet points in your head. That is totally cool for biology. All right, we're going on to the very last part of the exam, and it is 10.3. Describe the structure of ATP, adenosine triphosphate. Now, outline the named enzymes that break down and resynthesize ATP. So it's a 4 marker, this one, and you can kind of think of it, right? They want me to describe ATP, that's probably 2 marks. And then they want me to name the enzyme that breaks ATP down to ADP and PI, and then the enzyme that joins ADP and PI together to make ATP. And there's your 4 bad boy marks. Let's go. Well, ATP contains 3 phosphate groups. I'm just using the old simple diagram, the circles. You don't need to do a diagram, but it does help. If you draw it out, it will enable you to talk about all the different things in the structure. So it's got Three phosphate groups, what are they joined to? They're joined to a ribose sugar, not deoxyribose, but a ribose sugar. If you can't make your mind up, is it ribose, is it deoxy? Just say a pentose sugar because they're both contain 5 carbons. And then, of course, to a nitrogenous base. And where does the A come from in the abbreviation ATP? It comes from the adenine, which is the base. Now, you might get a bit messed up because the name of ATP is adenosine triphosphate. That base is not called adenosine. The base is called adenine. It just changes its name when it's joined to a sugar to adenosine. So anyhow, it's adenine, that base, and then we have ribose, and then we have the 3 phosphates chained together, and that it makes up our structure of ATP, and that's going to get us 2 beautiful marks. Now we're going to go on to say, well, how is this ATP broken down and how is it formed? So the bonds in ATP can be broken by a hydrolysis reaction. Again, they might ignore the name of the reaction because what they're really specifically asking for is the name of the enzyme that catalyzes that reaction, and that is known as ATP hydrolase. So ATP hydrolase helps with hydrolysis, breaking bonds in biological molecules. It's going to turn ATP to ADP plus PI, inorganic phosphate. And of course, ATP is synthesized with a condensation reaction using ADP, an inorganic phosphate, and catalyze that reaction is by the enzyme ATP synthase. So you've got ATP synthase and you've got ATP hydrolase. And of course, you could just to help yourself out, write the reaction out, which would be ATP, reversible symbol, goes to ADP, adenosine diphosphate, plus PI, which is an inorganic phosphate, and that's going to get us all the fat 4 marks, 2 for the description and 2 for the different enzymes. So yeah, we're going to cover it all. All right, guys, don't forget, sharing's caring. Hit the like, punch up the bell, subscribe. Let's get to the marks. I'll see you in the next video. Let's smash those exams. Don't forget, think about becoming a member.
You're still here? It's over.